So we already talked about bacterial shape, but another way to classify them is based on how they stain, particularly when doing a gram stain. And so this is telling us particular differences in their cell wall structure. And as you can see here, we have two different types in our pictures over here. We have ones that are looking purple and ones that are looking pink, all because of this gram stain process. So knowing what color they stain will tell you a little bit about their cell wall structure. So starting with our gram-positive bacteria, these are cells where the cell wall is high in peptidoglycan, and because of this, it's going to show purple. Then we have our gram-negative bacteria, and these cell walls have a thin layer of peptidoglycan with a polysaccharide layer on the outside, which means that it doesn't dye purple and is going to appear red. So I'm briefly going to describe what you saw in the gram staining process that we did earlier in the week. So here I have our two different cell types. As you can see, we have our positive cell and our negative cell, and they each have a slightly different cell wall structure. So how the staining process works is that we are first staining both cells purple. Even though the negative cells won't end up looking purple, they still receive the purple dye. So what happens is that you put dye on both of the cells, and you will stain it purple. So we're going to put purple dye on both of these, and that's the first step. But after this, we're going to use an alcohol wash, and what that's going to do is erase some of this top layer away, but this is still dyed purple, which means it's still going to appear purple. On our negative cell over here, when we use the alcohol wash and wash this layer away, that outside capsule layer, the polysaccharide, is what stained purple, not the inside. So now this one is still purple. And so when we go on to the next step, this one's undyed because we took that dyed layer away that was out here, and this side is still purple. So now that we go in with our last stain, the pink or the red stain, this one is still going to show purple because it's already been dyed. But when we come over here, this doesn't have any dye on it yet, so it's going to absorb the red or the pink dye, causing them to look different from each other, even though they both received the same stains. An easy way that I was taught to remember this is that I took this class, I took microbiology when I was at K-State. And if you don't happen to know, K-State's colors are purple. And if you're also unaware, K-State fans are not very fond of KU. So I was taught to remember that positive is purple for K-State and negative is red for KU. If that helps, sorry if you like KU. As we said at the beginning, only 1% of microorganisms are actually pathogens and cause disease to humans, which means that they have a lot of other functions and things that they can do for us, more than what we typically think of. So for humans in particular, we use bacteria for foods like yogurt and cheese and pickles. So all of these things are going through some sort of fermentation process where we are using bacteria to help grow these things. We also use bacteria to help us decompose matter. So whether that's us using it in some sort of compost pile, or this can be done on a larger scale as well. And very importantly, we use bacteria in every step of the nitrogen cycle. We talked about this in biology a little bit, the nitrogen cycle, but it is arguably the most important at converting atmospheric nitrogen into a usable form for plants. So I don't know how familiar you are with this, but plants in particular cannot use the atmosphere that's found, or the nitrogen that's found in the atmosphere. They have to first convert it into nitrates and ammonias, and they can't do that themselves, and so they have to rely on bacteria to do it for them, which is why in agriculture, one of the first things that we add to a crop is nitrogen, because they can't get that supply on their own. So if we look at uses in particular for humans, we have bacteria on our skin that help us to prevent infections. Good bacteria, there's hundreds and thousands of them living on you at all the time. There are bacteria in our guts that help digest our food. We cannot digest our food without them. And then if we look at it on a biotechnology level, we use them to make antibodies, insulin, the human growth hormone, vitamins, and many other drugs. So most people don't actually know this, but insulin in particular is a really good example, because as humans, we put human DNA into bacteria and have them grow insulin for us. So if you know any person who is diabetic, and most of the insulin that they are taking was actually grown inside a bacteria. So we are using them for our own good. The last general thing we have to talk about for bacteria is their sense of reproduction. And for asexual reproducers, we're talking about binary fission. So their nucleoid region, which is like their DNA, is going to be copied. Unicellular organisms will split in half, creating two identical bacteria. And that's key here for bacteria, that they are being made identical copies of each other. So if they're being made as identical, we need to think about how it is that we are getting any sort of genetic variation. And we know that we are, right? Otherwise, we wouldn't have any bacteria that are evolving and becoming resistant to our antibiotics, but yet we know that they are, which means they have to have variation in some form. So that can be from a variety of different ways. It can be from mutations that cause a random change in the genetic material, so it just happens by happenstance, no rhyme or reason. It can be through the process of conjugation, which is a DNA transfer between two cells. Two cells can go and decide to, shake hands and give each other DNA. Cool if humans could do it, but we can't. Then we have transformation, and again, this is similar, but it's the uptake of foreign DNA from an organism surrounding. So I like to think of transformation as like in a video game. Like if you kill somebody else, sorry, I'm thinking back to when I was like 8, but if you kill somebody else, you get to take all the weapons that they had. So if there's other stuff in your environment, in transformation, you get to pick it up. And then lastly, we have transduction. And transduction is when a virus transfers genes between prokaryotic organisms. So it's very similar to our conjugation, but here it's specifically talking about a virus between other prokaryotic organisms, not just any other prokaryotic organisms. So bacteria are really cool in this sense comparatively to humans, that they have all of these different ways, especially these three in particular, where they can share DNA with each other. So if you think about the fact that we have antibiotic resistance happening so fast, these are part of that, because they don't actually have to fully replicate and go through a generation to change their DNA. They can do that in their own lifetime, and humans don't have that ability.